I'm Oded Rehavi, a biologist from Tel Aviv University in the Department of Neurobiology, part of the Life Sciences faculty. And I've been studying many different topics. The main topic that we focus on is epigenetic inheritance in C. elegans nematodes.
Many different topics doesn't adequately encapsulate the diverse research interests of neuroscientist Oded Rehavi. After a year abroad as an art student in Paris, Oded decided to return to Israel to study neuroscience for his bachelor's degree. He's been at it ever since. Besides his work on epigenetics in nematode worms, Oded and his lab have also tackled a 2,000-year-old archaeological mystery, turned a notorious brain pathogen into a vehicle for delivering drugs to the central nervous system, and examined the rationale behind irrationality. Oded somehow also manages to find the time to run one of the most popular science social media accounts. Oded was elected an EMBO member in 2021, and we spoke with him this September. Welcome to the EMBO podcast. In the late 1940s, a Bedouin shepherd discovered the first of what would turn out to be over 25,000 text fragments of over 1,000 ancient manuscripts. The 2,000-year-old Dead Sea Scrolls found in the Qumran caves of the Judean desert are a biblical jigsaw puzzle. The scrolls were mainly written on the skins of domestic sheep, cows, and goats. Taking advantage of advances in ancient DNA sequencing and phylogenetic analysis technology, Oded led an effort to use genetics to help understand how to group and organize the scrolls, an effort that culminated in a highly cited 2020 research paper. Some molecular biologists might be tempted to see it as a story of science, solving a mystery that had long baffled the humanities. For Oded, it was a chance to learn and have some fun with a diverse group of colleagues.
So I learned so much from them, especially from Noam Mizrahi, who was our main collaborator, the biblical scholar on our team. And you can say like that we solved, we helped them do something that they couldn't do otherwise, but it really wasn't the case. I mean, that wasn't the motivation. The motivation was to do something interesting with colleagues that enrich you because they come from such a different place. And I think that's in many cases, that's the real motivation of what drives you. It's just sheer interest or that's the challenge of it. It's not necessarily solving a problem, at least for me. In that case, it was just fun. It was so much fun to just try this even that we decided to do it, and it stood. It worked out, and I hope that it was beneficial also for the understanding of the scrolls, that we added something. But the motivation was just having fun and talking to different people to really get out of your comfort zone. And this is, I think, how new things emerge. In my field, for example, transgenerational epigenetic inheritance, I feel that I'm becoming more and more limited because I'm already used to thinking about it in a certain way and think about the limitation and what's possible. And when it comes to a completely different field, that's where you have a lot more room to be creative.
That reminds me of a conversation I had a long time ago with Niles Eldridge, who had put forward punctuated equilibrium theory when he was a student with Stephen Jay Gould as his collaborator. And it was attacked very severely. And then it was not everyone, and it's by no means consensual, but it became part of the mainstream discussion. And years later, when he was already a senior scholar, we were having some caipirinhas in Brazil, and he got this very, very sort of disappointed, far away look, and he said, now I'm respectable.
I think this is very true also when it comes to transgenerational inheritance. It's not that there's no controversy about it whatsoever, but when we just started, that was the first paper that I published on it with Oliver Robert, my postdoc advisor. That was back then in 2011, that was completely new. And we got crazy emails from creationists and interesting things like that. And since then, since we and many other labs learned a lot about the mechanisms, I think that RNA inheritance in C. elegans is no longer controversial. Also because it's so easy to do. Anyone can just, feed worms on RNA, on double-strand RNA and get a transgenerational response. So that's very, very reproducible. So there's no controversy. The controversy now is whether this is also happening in other organisms. That's an open question. And there, as you know very well, it's still very, very controversial. But intellectually, I think for me, it's less interesting. We have a few model organisms. Maybe it happens, maybe it doesn't. Maybe it happens in humans. It's important, but it's less interesting, if you know what I mean. I think it's what will eventually decide how important this is. But the biology is there. It can happen. Before that, it was thought to be impossible, against the rules. And now we know that, at least in one of the main models of biology, it certainly happens.
Let's assume that it goes the other way. Let's assume that C. elegans, for example, is uniquely prone to transgenerational epigenetic inheritance. Do you think there's anything in the ecology and the biology of C. elegans in the wild that could justify a predisposition of this kind?
Yeah, we can speculate. We can speculate that there would be, because the generation time is so short. So the chances that the parent's environment will be relevant for the children's environment are high. Because they also don't go very far. So the chances are that the kids will experience the same environmental conditions, which is why it's worthwhile to prepare them. There's also, perhaps because there's less genetic variability between individuals coming from the same mother, more room for epigenetic variation. That's possible. However, that doesn't have to be the case, because some stresses or some environmental changes are very long lasting, and we, even as humans with a generation time of 20 years or more, still experience the same changes or the same environment that our parents did. This is true, for example, for some viruses that we encounter more over and over. In theory, it would be worthwhile to be prepared in advance. I'm not saying that it happens, but we can find challenges like this. But I think that if we even flip it again, that even if it was just a worm thing, a nematodes thing, and I don't believe this is the case, because I don't think we were that lucky. But even that, if that was the case, then 4 out of 5 animals on this planet are worms. So in terms of relevance, it's extremely relevant, just in terms of numbers of individuals. And if we think about the ecology, worms affect every process on Earth, the exchange of gases with the atmosphere, everything. So I think that it's possibly important, even if it's not conserved in humans or in mammals. But I think that, and this is just a guess, that even if the exact same mechanism isn't conserved, and this remains to be seen, it's too good of a trick to give up. That's what I believe. But I could be totally wrong.
Well, one of the fascinating aspects of your work is how regulated the process is, right? So this isn't it's taken on a very well-defined contour to the extent that you felt comfortable putting out rules for epigenetic inheritance in C. elegans. And one of them, I think the first one, is that the RNAs are very, very evenly distributed, so that you have very low variability in the progeny of a single worm. So just to give me an idea, how big is that progeny?
The average progeny is around 250 worms per mother. And first, in the first generation, there's really not much variability, but then there are mechanisms that intentionally introduce variability. Just like there are mechanisms for introducing genetic variability, recombination, and so on. Here, there are mechanisms that intentionally generate epigenetic variability, and we think of it, again, we could be wrong, as a sort of perhaps a bet hedging mechanism, where a few individuals will be more successful than the others because of these epigenetic changes. So there are rules. It's not just an epiphenomenon or a carryover of the parental responses, but we are just at the beginning of understanding them. We don't have a quantitative measurements of that. We don't know the stoichiometry of how much RNA is being inherited and how many small RNAs do you need per target, for example. These type of things, we are only at the beginning. And there are many open questions. And as you said, it's not so surprising now that there would be an epigenetic response propagating to the next generation. That won't be enough now to get you a paper in Cell, while it was a few years ago. Now you have to go beyond it, but there are a few open questions which are very, very big, like can epigenetic inheritance, which is typically transient, affect the process of evolution? When you think of that, it's something that takes a long time to happen. What type of responses can transmit transgenerationally? Can the brain coordinate these responses? And also many mechanistic questions of how exactly it works, which are still left open.
How representative do you think C. elegans is of this vast diversity of nematodes? How much do people look outside their worm?
So people look more and more. Also, when it comes to Canorhabditis species, people look more and more and more. That's now a part of many, many papers. And there are many similarities, but there are also very big differences between different worms. If you just look at or even nematodes, or even closely related Canorhabditis. For example, if you look at transgenerational inheritance, the mechanisms that exist in other related nematodes, closely related nematodes, you see that it changes very rapidly and evolves quickly. Many, even wild isolates of C. elegans, so it's still a C. elegans, many of them don't have RNA inheritance. RNA doesn't work. Now, if Sidney Brenner happened to pick a worm, not from Bristol, England, but from another place, from Hawaiian or something, and we worked with an Hawaiian strain, and maybe none of this would have been discovered. And part of the reason is that it's an arms race between the RNA system and all kinds of mobile elements that they defend against. So it's very fastly evolving. And I'm sure that the same is also true for the nervous system, which evolves fast. And comparing different nervous systems of related worms is a very promising field that is growing, and people are doing this. And I'm sure that it will discover fascinating things. But many of the things that were discovered in worms, in C. elegans, are also conserved in many other organisms. If you think about the Nobel Prizes, so RNAi and some of the mechanisms of cell death, this is all conserved. And also, if you look at the synapse and the genes that function in synapses in humans, they are all called, many of them are called UNC this and UNC that. UNC comes from uncoordinated. This is the original screen that Sidney Brenner did, where he mutagenized worms and looked for worms that move funny. And many of the worms that move funny, when he mapped and other people mapped the genes involved, were found to function in synapses and to be conserved in humans as well. And that's despite the fact that until recently, we didn't even know if worms have action potentials. So that's just very recently it was discovered that worms do have action potentials, but many of the neurons probably don't have them. And also the action potentials that they have are based on calcium.
In December of last year, Oded won the Schmidt Science Polymath Award. The Polymath Prize grants $500,000 a year for 5 years. The Polymath Award is not linked to any specific project, avoiding the usual gauntlet of milestones and deliverables, and allowing Oded and his team to explore new areas of research. I think one thing now that we are obligated to ask you in any interview is, so congratulations on the $2.5 million you got on this Polymath Prize, which everyone dreams about. What are you going to do with it?
Well, I'm checking many different directions. One of the things that we're more and more invested in now is mechanisms for controlling the duration of memory, even within the same generation. We have a very interesting phenomenon that we discovered that the lab is gravitating towards, and I hope to preprint it soon. But very broadly, we found that a very simple manipulation extends the duration of memories that are acquired within the same generation in C. elegans tenfold. That's and we are studying the mechanism. It's very, very interesting. So I'm putting more and more resources into that. But in addition, I dream of finding another model organisms for studying transgenerational inheritance, and preferably a very different organism. And I have a few candidates in mind that I want to develop. But we're also studying really, really different things in the lab. And the nice thing about this is that they encourage you, the Polymath Prize, Schmidt's Futures Foundation, they encourage you to try to do this, to take the risk. And even if it doesn't work, that's okay, as long as you tried. So I won't say I'm bored from what I'm doing now, but I really want to do new things to keep the fire going. I think that's the biggest challenge, to keep being really excited about it. It's not easy, especially during the pandemics, I think.
Yes, I guess it's an introspective, involuntarily introspective period, probably.
Right.
Also, one of the things that you're working on that I believe there's already been a preprint is this very interesting idea of, well, we can't get drugs in the brain, let's get a professional. And so you're mining toxoplasma as a delivery system, right, for brain therapeutics. How's that going?
That's going well. We pre-printed it a million years ago, perhaps in 2018 or something like that, or 19, I don't remember. The paper is under revision. I hope it will be published soon, but it's going well. What we did there is we used the fact that Toxoplasma gondii reaches the brain very effectively, also in humans, and can deliver its own proteins to the cytosol of infected neurons. It gets cleaned from the rest of the body by the immune system. So we hijacked a few secretion systems that it uses to do so, to deliver proteins of interest that are missing in certain neurological diseases. And in the preprint, we've shown that this is possible and it works well with a few proteins. As part of the revision, and it's not that there's been a million back and forth with the journal. In fact, it was just us sitting on it and doing more and more experiments. We show that it also works in mice very efficiently. We don't know how to work with mice. This was done with collaborators. And it works very nicely in mice. So that would be a part of the revision. And I think that really puts it in a new level because it works. I have to say that this project is now getting out of my lab, so I won't be working on it because we licensed the patent to a company that will try to actually translate it into a drug delivery platform. So once this is over, I won't work on it anymore. But I'm still very interested in symbiosis and using different pathogens or symbionts to do good things outside of Toxoplasma as well. So this is something that interests me a lot, and I hope to have additional ideas that I can test.
I get a little bit of a feeling from what you're saying that you may feel that it's time to move on to something heretical again. Do you have a sense of what that might be?
Yeah. So yes, it's everything that you just said. So combining experimental evolution with epigenetic inheritance, that's something that we're doing. We're also examining to what extent can the nervous system coordinate this. Is it enough to just think of something for a change to ensue? Think as worms think, not like we think. And also questioning some of the very basic assumptions regarding the mechanism. There are many assumptions made, many of them because things have been seen in other organisms. There's many studies, excellent studies on epigenetic inheritance in certain fungi, in plants, and some of the things that we think happen, we think because they were shown in other systems, but it's not entirely true. And also the phenomena that are being described are not exactly identical. So there are also very basic questions to ask about the mechanisms that we need to challenge, and also to examine other organisms, which is something we try to do a little bit. We don't really work with mice, but we try to examine it in other organisms that separated from the worms millions of years ago. But not only that, we also have other questions that we ask regarding the nature of memory, how memory is encoded. And again, C. elegans poses some difficulties to neuroscience. Some of the mechanisms that are missing in C. elegans, and still the worm can behave and learn certain things. And also the fact that we can't really understand its nervous system despite its simplicity. That's we have to be humble. Just 302 neurons, and still we don't really understand how it works. While in parallel, there are huge projects to solve the human brain and so on, which is much more complex.
Actually, that brings me to a question I'd very much like to hear your opinion on, which is So for C. elegans, as you say, you have 302 neurons. They're characterized. They have names. I guess people have their favorites and so forth. And not only that, and perhaps more importantly, you have a full wiring diagram of the brain. And of course, when you go to mammalian neuroscience or human neuroscience, there's a giant push to invest in building the full connectome or describing the full connectome of the brain. And at times it sounds a bit like the rhetoric around the Human Genome Project, which is we will understand physiology once we have the blueprint of a person. And as you're saying, for an organism that's a lot simpler and has a much smaller number of neurons and not to offend any worm, but a behaviorally less complex repertoire, perhaps. So if, from your point of view, if tomorrow we had a full wiring diagram of the human brain, for example, what would that change in our understanding of neuroscience?
So the connectome for the worm is very helpful. And some behaviors are understood in resolution that you can't reach in other organisms. And that also you see how certain things work and you truly understand them. And probably some of this could be done in higher organisms or more complicated organisms with more complicated brains. But it's certainly not enough. It's clear that a lot happens outside of the connectome or in parallel to the connectome, secretion of neuropeptides, many processes, and that we just can't, it's not enough to just look at the connectome and to understand what happens. And moreover, it changes all the time. And it's different between individuals. That will definitely be the case in humans, but it's also probably the case in C. elegans. We have a connectome of the worm for more than 30 years at an EM resolution. And that was a huge endeavor. No one wanted to do the second connectome because it's a lot of work. But recently, people did more EM reconstructions of additional connectomes. And you can see that they are a little different. And that's despite the fact that the worms are isogenic. And it was assumed to be completely hardwired. And these differences are also, it's another level of complexity. When it comes to the human brain, that continuously changes, that will be a big problem. So it would help. Some things will be learned for sure, but it won't be enough. It's not the human genome. The human genome was also not enough, but it's closer.
After a decade running his own lab and training 10 graduate students and postdocs in the process, Oded's anti-dogmatic views on transgenerational inheritance are now firmly within the mainstream.
I think it's evolution, and probably I'm the establishment. But I try to, I don't remember this exact quote, but I try to contradict myself, or at least to allow the students to contradict me. And I encourage them to do experiments without telling me. And then, to come once they already have some results. That's okay with me. And sometimes it works. And I'm sure that I'm the establishment when it comes to to transgenerational epigenetic inheritance. There I'm the establishment. In other fields, when we start a new project, I tell the students that they need to know the literature better than me after a year, something like that. And I think that this should be the model, that they are like independent researchers, and we provide the funding and some guidance with the experience that we acquire over the years. But they should be the new ideas should eventually come from them. I assume that we will become more and more boring as the years pass. As much as we'll try to fight it, it's inevitable.
So when you're recruiting new students or when they're coming to you, are they surprised by this approach? It used to be more common in some ways or in some places, but now it's becoming more and more that there's a preset project and there are time limits and all these things. How do they react, your candidates?
So I think they know this when they choose our lab. They know that it's a lab where you're supposed to take risks and some things might not work. They are aware, and also that I'm a little crazy in this aspect. But not all of them. Some of them need more guidance. Some of them want to do more, a safe project. What I try to do also is take people from very different fields. So now I have a student who just joined the lab, who's studied economy and worked in investing, but he's very smart. He's joining the lab. He's learning the biology now. I had some people from physics and math in the past, and I try to do this more and more. If I just recruit C. elegans researchers, they'll hit the ground running and they'll do great work, but it will bias us towards a certain direction. I try to take these students as well, or postdocs. They are amazing. I have a few joining now, but I also want to take very, very different people.
You mentioned earlier that one of your interests is in irrationality. Is this what your economist is going to be working on?
Yeah, actually, that's the case. Yeah, he's working on that, on economic irrationality.
In worms?
In worms. Yeah. We already had one paper published on that. It's I'm very proud of this paper. I think it's really cool. It was published in Nature Communication 2 years ago. And it's you can look it up. It's called Bounded Rationality in C. elegans nematodes, or something like that. More complicated name. And it's a collaboration with a neuroeconomist from the university, Dino Levy. It's one of the most fun projects I've had. We also worked on it for many, many years. And there, what we studied, people have shown, Kahneman and Tversky and others, that people act irrationally when it comes to the decisions that they make. For example, when you have to, you don't maximize the value of your decisions, of your choices. So if you and we wanted to, but normally the explanation, and this Kahneman, Tversky got the Nobel Prize for that. Kahneman got the Nobel Prize for that. Tversky unfortunately died before he would have won the Nobel Prize. But most of the explanations come from the realm of psychology. So you don't do it because of emotions or regret or things like that. We wanted to understand the underlying biology. So we studied whether worms make irrational decisions. And what we chose and what the student that joined will do other things. Guy, we will study other paradigms. What we showed in that paper is that we studied a particular paradigm that's called independence of irrelevant alternatives. What it means is that, let's say you have a preference. You prefer oranges over apples. Okay? You like the taste of oranges better than you like the taste of apples. It turns out that in certain situations, if I add another fruit, let's say pears, suddenly it would change your preference. You'll prefer apples over oranges. And this has been shown in humans, studied forever. There are many, many studies on this. It wasn't clear about why this is the case. So we wanted to do the exact same thing in C. elegans. So we let the worms choose between two different orders in the presence or absence of another order. They just did the same thing. And the worms do chemotaxis. They move towards this order. And the power of C. elegans is that we know for each order which individual neuron or pair of neurons sense it. So we can see how the decisions that the worm make are influenced by the involvement of different circuits or by the activations of different neurons in the circuit. And what we found when we did these experiments, it's really remarkable in my opinion. We found that the worms, most of the time, they behave very, very rationally. So they prefer A over B, even in the presence of C, even when you increase the concentration of C and C and make it more and more attractive. That is true for most of the cases. However, in certain situations, the addition of C will make them switch and they'll prefer B over A. And this happens when C takes more resources from the sensations of order A than from the sensation of order B. It's a very simple clash. And we can make genetically engineered worms that are more or less rational by changing the resources that we allocate to the sensation of each order. And this parallels things that have been observed with regard to other irrationalities, or quote unquote, to other illusions, for example, to visual illusions. Some visual illusions stem from the exact same principle. So we think that in certain situations, bad decisions or irrational decisions could be stemming just because of limitations of the nervous system, of how it calculates, or from boundaries of neurons and how they fire. You don't need to involve emotions or any higher cognitive functions. It could just be the limitations of how neuron works.
So it sounds like it's rational at the cell circuit level, but irrational at the organismal level. It's a resource allocation problem being solved by cells.
Yeah. And there could also be a reason for that. What appears to be a reason could actually be, what appears to be a bug could be a feature. In certain cases, this is not the case, but it's clear that some of the bad decisions that we make result from heuristics that we take, from limitations, for wanting to make a decision fast. Because it's a more common stimuli that you will encounter, not necessarily because you don't have to maximize all the value in the world and take everything into consideration. But it's nice because in Silence you can actually ask these questions and see it at a neuronal and mechanistic level.
And finally, of course, we also have to discuss a little bit your other life as a social media star. So. And I noticed that right before we had this conversation, you put up a Joe Pesci meme, the famous "Am I a clown? Am I here to amuse you?" So that has a little bit of an edge, no, in the term. So does that reflect any level of negative feedback on this participation?
Yeah. So I started with Twitter, just for fun. It wasn't strategic at all. And I enjoy it. And when I see a good meme or a video or something funny, I have to caption it. I can't control it. It's an addiction. I have to do it. And I do it very, fast without thinking so much. So I often also erase tweets that I think I shouldn't have tweeted. But, and it became a thing and many people see it. And I get many good things out of it, good feedback. I learned I met you and we interacted. That's fun. And also Twitter, there are many good aspects of Twitter, but I also think that it probably reflects badly on me sometimes because scientists likes to feel respectable and serious. And I have many agendas in my tweets. I make fun of peer review. I make fun of all kinds of things, but the overall theme, the things that I care about the most is battling against the pomposity of scientists and taking yourself too serious. And I think that it's very important because it's a shame that we limit ourselves like that, that we have to be serious all the time. And I think you can be not serious, you can make jokes and criticize the system and still have serious research. And I think that, but I'm sure not everyone sees it that way. And I'm sure that in certain situations where I was appraised, or in certain evaluation committees, someone raised an eyebrow and said, this person, we don't need this sort of type of clown around us or something like this. I'm sure that happened, but I can live with that. And I think that the good overweights the bad, and that I get more out of it than what I lose. But there is a price. And you have to be careful on Twitter not to upset anyone. And this can happen because of nonsense, as you know. But so I do it. I can't control it. I try to limit myself. I also, had an exchange with you about how to do it. It's very, very difficult. It's almost impossible. But I looked at my analytics yesterday. I don't do this often. And I saw that I'm really, losing it less. I'm using it less. I'm down 25% or something like this. So I'm proud of myself. Let's see how long it lasts.
It's interesting that it seems to have been the social media platform that took off for scientists, right? I don't know if it's because of the format itself, that it's more punchy and to the point, and somehow, for me at least, I don't know if this has been your experience, but I don't use other social media platforms anymore, but it was much easier to structure a community. Like for example, I haven't used Facebook in a decade, but when I did, the sort of crazy comments from someone's racist uncle would eventually filter in, and that doesn't happen on Twitter. At least on science Twitter. I know that there are other problems. It seems to be a community that's very geared towards support and helping people, and you see people beginning projects together, sharing papers, sharing reagents. It's for all the toxicity of social media, it seems to have a very strangely positive dynamic with the scientific community.
I agree. I also didn't use Facebook. Never. I was on it, but never used it. And I share your feeling that it's mostly positive. And I don't know why it catch so well with the scientific community, but it did. And most of the exchange is supportive and positive. Sometime, every once in a while, someone gets in the middle of a controversy. Sometimes for no good reason, but most of the times they said something that they shouldn't have said. So it's self-correcting. But I do think we live in a bubble, and in some corners of Twitter, there's very bad things going on. I wonder how you deal with it, actually, because I try to avoid COVID as much as I can. I want to say things about it, and I'm on the side of enlightenment in the thing that I think about COVID, although I'm not an immunologist, but obviously, I have the right thoughts that would be approved by our community or the people that we interact with on Twitter. But I know that people get into a lot of trouble clashing with anti-vaxxers and things like that. I tried it a little bit. That was in Hebrew, and that was really violent. And I promised myself that I will just avoid it, because, and this is the part where the bubble that we create bursts, and we experience the real world. And I don't know if you experienced it, but I see it around me all the time.
I think earlier in the pandemic, I experienced more of it, particularly when I was doing the updates for Nature Medicine. But I developed a few working principles, which I don't think are particularly creative. But one of them is, the thing to understand about these social media platforms is that it's an attention economy, right? And this is really hard to explain to people when they're new, but to say, if you're drawing attention to something, it doesn't matter if you're making a negative comment about it, if you're saying this is wrong. If you put this person in the spotlight, the algorithm pushes them up, right? So if a bit of disinformation, and some people engage directly, and I see some accounts, doing valiant battle, and sometimes getting very, very bad vitriolic comments and sometimes threats. And of course, it's always worse with women when they engage, and there are several ugly aspects to this. So for me, the way to go is, if I see a claim that is wrong on some new medications, I don't engage with the person making the claim. I put up the right information, right? And if that's trending, I try as much as possible not to, I don't know if in the long run this is good or not, but it has worked for me. Instead of directly rebutting some piece of misinformation, I'll go and put up, what it seems to be the correct or the best available information on the topic at the time, right? So to stay, I'm not anyone who knows me. I'm not a particularly positive person in real life, but in this kind of thing, it pays more to put out the right information than to boost the wrong information, which is really counterintuitive, but it's what you're doing. I mean, every once in a while there is a slam dunk takedown, but it's very rare, right? And it works even when it's referring to things that are outside of the social media, that are in traditional, for example, journal publication. In this conspiracy universe, when a paper is retracted because it was wrong, because maybe, Mentos in Coca-Cola does not cure acute COVID or something, that becomes part of the conspiracy narrative, right? That becomes, big something had it withdrawn from journal X, Y, or Z. So it's very hard to engage directly with misinformation and not push it up.
Yeah, and you don't know what would work. So, for example, I think that people celebrated the novelty of the mRNA vaccines because they wanted to celebrate, the progress in science and how amazing it is that they did the vaccine so well and so fast. But that was spun into something bad by the people that say that it is novel, so it's untested and so on, which is not true. But once you started it, it's impossible. So you don't know how it will turn out.
Yeah, and it's clearly had an impact in some places much more than others, and it also has to do with just the general level different societies have of trust. And it's not always obvious what those will be. So in corresponding with friends in Japan, for example, I found out that the degree of overall vaccine hesitancy or skepticism over there long preceded COVID and made huge dents in things like the HPV vaccine. So there was sort of a sustained problem. And it's also difficult, I think, for the scientific community to engage with this sort of thing because there's a misconception on our part that this is happening because of a lack of information. And it's actually because of a presence of bad information. So when you get one of these anti-vax guys, one of these anti-vax people, normally they are not in an information vacuum. They have an incredibly rich, sophisticated literature. They spend more time on this probably than the person who just goes out and gets vaccinated. And they cherry pick peer-reviewed studies. There's always an engineer or another who is going to show you how the biology is all wrong because, and then there's that tempting, once it's math, it's magically objective kind of aspect of discourse. But it's not this kind of thing, I think this is very difficult for us to accept, that you're coming in and bringing the knowledge, right? There is a presence of bad knowledge, and then that's really different, right? Because people invested in that a lot, they read a lot, they write a lot, they have their own groups, so it's difficult to get in that. And the other thing for me with COVID that was extremely important is I'm a biologist, and this was my rule, and this was something where I called people that I knew who were at one time or another unwittingly violating it, which is say, if you're not a doctor, don't give medical advice, right? And I would get messages from people who say, I have this or that condition, I have this autoimmune disease, should I take this? And I would simply refuse to answer very politely. Decline, say, look, I am not a doctor. I cannot give you advice on what you should go to the pharmacy and take.
Right, but many times the questions that they ask is, do I get vaccinated? And there you're willing to say yes.
Yes, but so I don't get a lot of those, but I get more of when people write, I have this specific condition. Mm-hmm. Right? Should I get vaccinated? Uh-huh. Or I'm under chemo or radiotherapy of this kind. Should I get vaccinated? And then it's not a linear answer because, for example, some kinds of live attenuated vaccines are not a good idea if you're in a particularly immune suppressor vulnerable mm-hmm. state. And this long precedes COVID, right? And there are best practices. And you don't know about each. I mean, medicine is an individual thing, right? You have to know about the person in front of you.
And you also say, people say it's your responsibility to talk about COVID because the information, you have to put the right information out there. But as you said, it's not a problem of there's no information. It's the problem that there's bad information there. So the question of whether should I get involved in it or not is not so simple. And I learned that some topics it's extremely difficult to discuss on Twitter. It's not a great platform for discussing anything. So I won't touch politics at all, although I'm a liberal and left but I won't do it because, like you said, it puts someone in a spotlight to now make a big deal out of something and attack you. So I know the limitations of the platform, and I decided not too long ago, to narrow what I discuss and what I don't. It's just too much to lose, and it's not a real good platform for debating and discussing any topic in the world.
No, I do think it's been interesting to see some people sort of pushing the envelope. So, for example, Florian Kramer at Mount Sinai had something like 140 tweet thread explaining pretty much everything about the vaccines. And it was a masterpiece. It was as good as any review.
And who reached tweet number 140? It's really challenging.
A lot of people, because you can see the likes on each tweet in the thread, right? So this sort of magnum opus went viral. And you do see, in terms of interdisciplinarity, you see people talking who probably normally wouldn't, that are connected through different things. You see people interacting with the funding agencies and the journals, and it's great because this is happening in an open forum, right? So you do get to see interactions that would be closed. And everybody, as you said, tries to be on their best behavior, which I think is great for everybody. I mean, there are histories of, I'm sure you have your own, of people who have received unpleasant messages from somebody who shouldn't have sent it, either from a funding agency or from a journal or from a reviewer for something like that. Whereas out here, we're all trying to be civilized. One of the things I know you mentioned when you were interviewed by the EMBO communications team was that you would like to use this membership to organize more interdisciplinary meetings. And I'm curious if you have a topic in mind of what would make for a good interdisciplinary meeting right now.
Yeah, I think that, as you know, we organized just before the pandemic, the Physiologically Irrelevant Conference, the Woodstock of Biology, as it was later dubbed. And there, what worked well is that there wasn't any plan. There wasn't any topic. It was truly interdisciplinary in the way, because you can just present whatever you want. If you want to come there and present something about biology, and it sounds perhaps too wild, but too unorganized or unstructured, but it worked. I think that I can probably come up with more narrow topics for an interdisciplinary meeting. But perhaps this is something that can be just, emerged bottom up without too much planning. Especially if you build it around a community where the science is excellent, like the EMBO community. There are so many different topics studied, and everyone is a good scientist. So if you use this as the backbone and build around it, I think something interesting can come up.
That tracks well with your description of how the Dead Sea Scrolls project began. That you were, I think in one podcast, you said you got on a bus and 8 years later you were still on a bus.
Yeah, that's exactly how it happened. There was no planning involved.